Hello and welcome to another video lecture. आपके syllabus के according we are supposed to discuss about the absorption of carbohydrates, proteins and lipids. इस lecture में we will try to understand about the absorption of carbohydrates. Absorption, as you know, is the movement of these simple diffusible molecules from the lumen of the intestine into the blood. For example, here, if this is the intestinal lumen and present in this intestinal lumen, we have these simple diffusible molecules. Diffusible molecules are those molecules which can easily cross the plasma membrane of the cell present on this intestinal wall and will enter into the blood or the capillaries. So this movement from the luminal side of this intestine through the intestinal wall and entry into the blood of the organism is the absorption. And remember, for this absorption, these molecules must be in the most simplest form, most diffusible form, so that they can easily diffuse through the plasma membrane of the cell. So this is the absorption of these molecules. With respect to this absorption, we have two major classes in the carbohydrates. One class of carbohydrates requires the enzymatic digestion before it is being absorbed, and another class of carbohydrates, they don't require this enzymatic digestion, or you can say carbohydrates, when they are very simple structurally, they can be easily absorbed without being digested, without being further broken down by the enzymes. These carbohydrates which can be absorbed without being enzymatically digested, though they are called as the monomers. For example, we have this glucose or fructose. These monomers are ingested and absorbed as such. They don't require the digestion. But some carbohydrates are very complex structurally. For example, we have this glycogen or this starch. These carbohydrates, when ingested, they require the enzymatic digestion. These complex structures of these carbohydrates, they are being first enzymatically broken down into most simpler forms. And when they are being broken down by enzymes, they are ultimately, finally converted into the monomers. These carbohydrates are finally converted into these monomers, these glucose, galactose and fructose. And it is in this form, these monomeric form, they are being absorbed, or you can say the absorption of glucose, fructose and galactose is actually the absorption of carbohydrates. So when we ingest these carbohydrates as the glycogen or the starch, these are polysaccharides, when we ingest these polysaccharides, these complex carbohydrates, they are first enzymatically digested, they are first enzymatically converted into the disaccharides or limit dextrinase. And as you know, we have two sets of enzymes for this. One is the salivary amylase, and then we have the pancreatic amylase. So you can say these amylases convert these complex carbohydrates or polysaccharides into the disaccharides or limit dextrinase. So we have these amylase products as the maltose, sucrose and lactose. So ये amylases क्या करेंगे, ये complex polysaccharides को convert करेंगे into the disaccharides और limit dextrinase. और इसके लिए हमें क्या हमारे पास है, एक तो salivary amylase है, एक pancreatic amylase है. ये आपने already digestive system में जो already पढ़ा होगा. यहाँ पर थोड़ा सा ब्रीफ डिस्क्रिप्शन देंगे ताकि हमें एब्जॉर्प्शन समझने में थोड़ी बहुत आसानी होगी. तो ये कॉम्प्लेक्स कार्बोहाइड्रेट्स को पहले हमने एंजाइमेटिकली ब्रेक डाउन करना है इनटू दीज मोनोमर्स. अगर हमने ऑलरेडी इन कार्बोहाइड्रेट्स को एज ए मोनोमर इंजेस्ट कर लिया तो हमें एंजाइम डाइजेशन की ज़रूरत नहीं है. फॉर एग्जांपल, इफ़ वी इंजेस्ट द ग्लूकोज को फर्दर ब्रेक डाउन करने की ज़रूरत नहीं है. लेकिन अगर हमने पॉलीसेकेराइड्स लिए हैं लाइक ग्लाइकोजन और स्टार्च, उसको एंजाइमेटिकली ब्रेक डाउन करना है पहले इन दीज मोनोमर्स. सो ये एंजाइमेटिकली ब्रेक डाउन जो होता है ये डाइजेस्टिव सिस्टम में होता है. पहले हमारे पास है सिल्वरी अमाइलेज और पैंक्रियाटिक अमाइलेज. दे विल ब्रेक डाउन दीज कॉम्प्लेक्स पॉलीसेकेराइड्स इनटू दीज डाइसेकेराइड्स. और ये जो अमाइलेज एक्शन होता है, इट ऑकर्स विद इन द ल्यूमिन ऑफ दिस digestive tract. We can say it occurs within the lumen of the intestine, because these amylases, they are released by these glands into the lumen of the intestine or into the lumen of the mouth. So यहाँ पर lumen में digestion होती है, luminal digestion. लेकिन जो ये डाइसैकेराइड्स एक बार इस पॉलीसैकेराइड्स से हमारे पास आ गए, नाउ दीज डाइसैकेराइड्स will be converted into the monosaccharides, and these disaccharides will be converted into the monosaccharides by various disaccharidases. We have some enzymes. These enzymes will convert these disaccharides into the monosaccharides, and these enzymes, they are called as the disaccharidases. Like we have lactase, we have sucrase. These are the enzymes present in the plasma membrane as the integral membrane proteins on the cells present on this intestinal wall. That is, these enzymes, they are not being released into the lumen of the intestine. They are present on these plasma membrane as a integral membrane proteins. And on the surface of these plasma membranes, these disaccharides, they are converted into the monosaccharides. We have these glucose, galactose and fructose. For example, this maltose will be converted into 2 glucose molecules by the maltase enzyme. This maltase is a disaccharidase. Similarly, we have this sucrose. Sucrose will be converted into the fructose and the glucose by sucrase disaccharidase enzyme. And we have this lactose. It will be converted into the galactose and glucose by the enzyme lactase. So these disaccharidases will convert these disaccharides into these monosaccharides. And remember, this conversion is necessary for those polysaccharides whose structure is very complex, which cannot be absorbed as such. For example, as I said, these polysaccharides like starch and glycogen. In case of simple carbohydrates, we don't need this enzymatic digestion. Now we have monomers, these glucose, galactose and fructose. These 3 monomers are present in the lumen of the intestine. For example, here in this case we have these 3 monomers present in the lumen of the intestine. As I said earlier, this absorption involves the movement of these diffusible simple molecules through the wall of the intestine and enter into the blood of these capillaries. Now, how these simple diffusible molecules cross this wall of this intestine? As I said, on this wall we have these cells. These are the cells present on the wall of this intestine, and these 3 monomers, this glucose, galactose and fructose, they are present on this luminal side. Here you have the luminal side of the intestine. For example, this is the glucose molecule, this is the galactose molecule, and present here is the fructose molecule. Now we have these monomers, this glucose, galactose and fructose present in the lumen of the intestine. Now let us try to understand how these monomers will cross this wall of the intestine and will enter into the blood of the capillaries. This is what absorption is. So absorption में हमने क्या करना है कि इस movement को जो from the lumen of the intestine towards the blood of the capillaries है उसको समझना है. So हमें ये देखना है कि ये जो मोनोमर्स हैं, ये जो ग्लूकोज है, गैलेक्टोस है या फ्रुक्टोस है, जो हमारे पास एंजाइमेटिकली डाइजेशन से हमारे पास आ गए या फिर हमने एज सच इनको इंजेस्ट कर लिया. नाउ दे आर प्रेजेंट इन द ल्यूमेन ऑफ दिस इंटेस्टाइन. हियर यू कैन सी दीज सेल्स, दे आर प्रेजेंट ऑन द वॉल ऑफ द इंटेस्टाइन. यहाँ पर 2-3 चीजें आपको समझनी है कि ऑन द वॉल ऑफ द इंटेस्टाइन दीज सेल्स आर इंटरकनेक्टेड विद ईच अदर थ्रू दीज टाइट जंक्शंस. These tight junctions will seal the passage between the two cells. Intercellular space जो होती है उसको ये seal कर लेते हैं. Now these monomers cannot cross this barrier through this passage. This passage is sealed now. Here we have this tight junction. और ये tight junctions का काम क्या होता है कि intercellular space के बीच जो passage है उसको seal कर लेते हैं. Now these monomers cannot cross this. तो ये मोनोमर कैसे क्रॉस कर लेंगे? इनको क्रॉस करना है थ्रू दिस प्लाज्मा मेम्ब्रेन. यानी आप कह सकते हैं एक सेल से दूसरे साइड में जाना है, दे हैव टू क्रॉस दिस बैरियर थ्रू द सेल प्रेजेंट ऑन दिस इंटेस्टाइनल वॉल। अब ये इस सेल को कैसे क्रॉस कर लेते हैं? एज यू कैन सी हियर वी हैव टू साइड्स ऑफ द सेल। Plasma membrane of this cell which faces the lumen of the intestine is called as the apical membrane. This side of this membrane, this plasma membrane which is towards the lumen of the intestine is the apical membrane, and the side of the cell which is away from the lumen of the intestine is called as the basolateral membrane. So we have this basolateral membrane and we have this apical membrane. This cell is polarized in this sense that we have different set of transporter proteins or integral membrane proteins on the basolateral side, and we have different set of proteins or integral membrane proteins on the apical membrane side. Or you can say towards the luminal side we have different set of proteins present on this plasma membrane, and towards the basolateral side we have different set of proteins. In this sense it is the polarizer, and this polarization is created by these tight junctions. ये अलग topic है, we are not supposed to discuss it here. Here simply you remember that we have different set of transporter proteins present towards the luminal side, and we have different set of transporter proteins towards this basolateral side. Towards the apical side we have this sodium glucose transporter and we have this glucose transporter 5. These two transporters play very important role so far as the absorption of these carbohydrates is concerned. So you have to remember them. So you have to remember this sodium glucose transporter and glucose transporter 5, and you have to remember that these two transporters are present towards the luminal side, or you can say towards the luminal side of the intestine. And towards the basolateral side we have this sodium potassium pump, and then we have this glucose transporter type 2. So glucose transporter type 2 is towards the basolateral side, and glucose transporter type 5 is towards the apical side of this cell, towards the luminal side. So in order to understand the absorption of these carbohydrates, you have to remember 4 membrane transporters. One is the sodium potassium pump, glucose transporter 2, which are towards the basolateral side, and then we have the sodium glucose transporter and glucose transporter 5, which are towards the apical side or towards the lumen of the intestine. Then you have to remember these tight junctions which seals the space between these two cells, and they don't allow these monomers to cross this barrier through this intercellular space. So these tight junctions not only seal the space, they also create the polarity with respect to the distribution of these membrane proteins. Now let us try to understand how these monomers will cross this barrier. As I said, this barrier here, this option is sealed by the tight junctions. Now to cross this barrier they have to cross this plasma membrane at 2 sites, first at this apical side, then at the basolateral side. So the absorption of these monomers, this glucose, galactose and fructose, occurs through 2 steps. In the first step they cross this apical membrane and they will get transported from the luminal side towards the inside of the cell. This is the first step. That is, in the first step these monomers, they will cross this apical membrane and will enter into the cell. And in this step we have 2 transporters. To perform this first step we have 2 transporters. One is this sodium glucose transporter, then we have this glucose transporter 5. Through this sodium glucose transporter, this glucose and galactose gets transported, or you can say this sodium glucose transporter transports the glucose and galactose. This sodium glucose transporter cannot transport the fructose. This fructose is having separate transporter. And remember, this transporter, they are integral membrane proteins. ये proteins होते हैं plasma membrane पर. So we have one set of proteins acting as the transporter for the glucose and galactose. We have separate membrane transporter for the fructose. This fructose cannot transport through this sodium glucose transporter. So here you can say glucose and this galactose gets transported through this sodium glucose transporter. We have single transporter for both glucose and galactose. So this will cross this apical side of the membrane, apical membrane of this cell present on the wall of the intestine, and will enter into the cell and will enter inside the cell. And remember, for these two monomers, we have single transporter present on the apical membrane. And for this fructose, we have separate transporter. And this transporter will cross this fructose from the luminal side and will enter into the cell. Now we have fructose, galactose and glucose molecule within the cell. All these monomers, once present inside the cell, they cross this basolateral membrane through the same transporter. This transporter, glucose transporter second. You can say that we have two separate transporters on the apical side, on the apical membrane. These two transporters transport these glucose and galactose and fructose. And we have single transporter on the basolateral side which transports all these monomers from inside of the cell towards the blood, towards the capillaries. Here you can say that we have two separate transporters on the apical membrane. One transporter specifically for the glucose and galactose. Then we have separate transporter for the fructose. Once inside the cell, all these three monomers can cross this basolateral membrane through the single transporter. And this single transporter is glucose transporter type second. This glucose transporter 5 present on the apical membrane transports this fructose. So this is the first step involves the transfer of this apical membrane towards the inside of the cell, and from inside towards the capillaries it occurs through the single transporter, glucose transporter second. You have to remember that we have two separate transporters for these three monomers. Second point you have to remember is that the transfer of this fructose from the luminal side towards the inside of the cell is facilitated diffusion. This is facilitated diffusion. It does not require any energy. So it depends upon the concentration difference and it is facilitated by this transporter, this glucose transporter 5. And the transfer of all these monomers through this glucose transporter 2 is also the facilitated diffusion. So we have facilitated diffusion occurring at two sites through two transporters. One is this glucose transporter type 5, which transports the fructose from the luminal side towards the inside of the cell. Then we have the transport through this glucose transporter type second through this basolateral membrane, and it transports three monomers, these glucose, galactose and fructose from inside of the cell towards the blood capillaries, and this occurs through the facilitated diffusion. आपको ये याद रखना है कि एक तो facilitated diffusion जो है it occurs through the glucose transporter type 5 and it involves the transport of this fructose from the luminal side towards inside of the cell. और दूसरा facilitated diffusion जो होता है वो basolateral membrane पे होता है through this glucose transporter type second, and it involves the movement of this glucose, galactose and fructose from inside of the cell towards the blood capillaries, and it also occurs through this facilitated diffusion. It does not involve the expenditure of the energy. So in this facilitated diffusion we have the involvement of two transporters, two glucose transporters type 5 and type second. One is present on the apical side and another is present on the basolateral side. But the transport of this glucose and galactose is not so simple. So to understand it, we have to understand the functioning of this sodium potassium pump. This transport of the glucose and galactose is secondary active transport. How it occurs? It depends upon the concentration of sodium ions on two sides. and it involves the expenditure of energy. It is active transport and it occurs against the concentration gradient. For example, we have the glucose concentration present in the luminal side is less as compared to inside of the cell, so it is against the concentration gradient. As you know, to move the molecule against the concentration gradient, you have to spend energy. So how it occurs, let us try to understand this. First, you have to understand the sodium potassium pump. What it does, it moves sodium ions. We have the sodium ions present on this side. It will move these sodium ions towards the outside. That is the sodium potassium pump. It moves 3 sodium ions from inside of the cell towards the outside of the cell, and simultaneously it will move 2 potassium ions from outside towards the inside. This functioning of sodium potassium pump involves the movement of sodium and potassium ions. This sodium potassium pump, with the expenditure of a single ATP molecule, moves these 3 sodium ions from inside of the cell towards the outside, and simultaneously it moves 2 potassium ions from outside to inside of the cell. And in this transport, in the transport of these 3 sodium and potassium ions per cycle, it utilizes 1 ATP molecule. This is the active, primary active transport, because it directly utilizes 1 ATP molecule in this cycle of this transport. This transport occurs at the basolateral side. यानी basolateral side में जो ये सोडियम पोटेशियम पंप है, ये क्या करता है, सेल के इनसाइड में से 3 Na positives को मूव करता है टुवर्ड्स द आउटसाइड ऑफ द सेल. अब यहां से जो ये Na पॉजिटिव मूव करेगा टुवर्ड्स द आउटसाइड ऑफ द सेल, इससे क्या होगा, इससे ये होगा कि इनसाइड द सेल द Na पॉजिटिव कंसंट्रेशन विल डिक्रीज. यानी अब हमारे पास क्या है कि सेल के अंदर Na पॉजिटिव कंसंट्रेशन कम है एज कंपेयर टू आउटसाइड ऑफ द सेल. हियर यू कैन से दिस इज द आउटसाइड ऑफ द सेल. यहां पर कंसंट्रेशन कम हो रही है सोडियम आयंस की. व्हाई? बिकॉज दिस सोडियम पोटेशियम पंप इज मूविंग दी सोडियम आयंस टुवर्ड्स द आउटसाइड. अब यहां पर एक कंसंट्रेशन ग्रेडियंट क्रिएट होता है. इस साइड में हमारे पास सोडियम आयन की कंसंट्रेशन ज्यादा है और इस साइड में सोडियम आयन की कंसंट्रेशन कम है. ठीक है. नाउ वी हैव टुवर्ड्स द ल्यूमिनल साइड वी हैव मोर ionic concentration of the sodium ions and we have less ionic concentration inside the cell. Now what will happen, these sodium ions will be having natural tendency to move from this higher concentration towards the lower concentration. यानी ल्यूमिनल साइड में जो कंसंट्रेशन हमें ज्यादा मिल रही है सोडियम आयंस की, वहां से ये सोडियम आयंस मूव करेंगे टुवर्ड्स द लोअर कंसंट्रेशन. अब ये लोअर कंसंट्रेशन सेल्स में क्यों है? Because we have this sodium potassium pump at the basolateral side which is moving these sodium ions from inside the cell towards outside of the cell. यानी सोडियम पोटेशियम पंप यहां पर सोडियम आयन की कंसंट्रेशन कम करता है और यहां पर जब सोडियम आयन की कंसंट्रेशन कम हो रही है तो ऑब्वियसली यहां पर ये ज्यादा है एज कंपेयर टू इनसाइड ऑफ द सेल. नाउ दीज सोडियम आयंस विल मूव फ्रॉम आउटसाइड टू फ्रॉम ल्यूमिनल साइड टुवर्ड्स द सेल. लेकिन ये सोडियम आयंस जो होते हैं ये मूव करते हैं थ्रू दिस ट्रांसपोर्टर, थ्रू दिस सोडियम ग्लूकोज ट्रांसपोर्टर. They cannot cross this plasma membrane at this side. They will move, they will cross this plasma membrane only through these transporters. Now this sodium ion will bind with this transporter and simultaneously we have the binding of this glucose molecules to this transporter. यानी असल में मूव करना है किसको? सोडियम आयन को. बिकॉज वी हैव डिफरेंट कंसंट्रेशन ग्रेडियंट. यहां पर सोडियम की कंसंट्रेशन ज्यादा है और यहां पर कम है. लेकिन इस ट्रांसपोर्टर के साथ उसी वक्त ये ग्लूकोज मॉलिक्यूल भी बाइंड होते हैं. अब ये इसको हम कहते हैं को ट्रांसपोर्टर बिकॉज इट इज ट्रांसपोर्टिंग सोडियम एज वेल एज ग्लूकोज आयंस. And it is symporter, it transports them in the same direction. असल में मूव होना है सोडियम आयन को डाउन द कंसंट्रेशन ग्रेडियंट मेंटेन्ड बाय दीज सोडियम पोटेशियम पंप्स. अब जो ये सोडियम आयन्स मूव होते हैं फ्रॉम द ल्यूमिनल साइड टुवर्ड्स द inside of the cell, simultaneously these glucose molecules get bind with this transporters and they also move along with these sodium ions from the luminal side towards inside of the cell. Or you can say movement of these glucose molecules depends upon the movement of these sodium ions. If sodium ion concentration is not higher in the luminal side, it will definitely affect the movement of glucose molecules. These glucose molecules will move from the luminal side towards inside of the cell only when the sodium ion concentration in this side is higher. And this side it is higher only because of the action of sodium potassium pump present in the basolateral side. As energy is spent here you have the utilization of ATP molecules. At this side you have the utilization of ATP molecules. As I said, in per cycle of this ionic movement we have one ATP molecule consumption. But here it is not the expenditure of ATP molecule directly, but it is using the concentration gradient of these sodium ions maintained by this sodium potassium pump for the transport of glucose molecules. So that is why this transport by this sodium glucose transporter is called as secondary active transport, because it is not directly utilizing the ATP molecule, it is utilizing the concentration gradient of these sodium ions for the glucose transporter. And this concentration gradient of this sodium ions is maintained by this sodium potassium pump, and this sodium potassium pump is directly utilizing the ATP molecules. This is the primary active transport and this is the secondary active transport. यानी जो concentration gradient यहाँ पर सोडियम आयंस का मेंटेन किया है सोडियम पोटेशियम पंप ने बाय डायरेक्टली यूजिंग दिस एटीपी मॉलिक्यूल, उस सोडियम ग्रेडियंट को ये ट्रांसपोर्टर हाईजैक करता है यानी यूटिलाइज करता है फॉर द ट्रांसपोर्ट ऑफ ग्लूकोज मॉलिक्यूल फ्रॉम द ल्यूमिनल साइड टुवर्ड्स इनसाइड ऑफ द सेल. So this is the transport of these monomers. In short, you have to remember there's this glucose and galactose transport from the luminal side towards inside of the cell is a secondary active transport. This movement, the movement of this fructose does not depend upon the sodium ion concentration. It is the facilitated diffusion. It depends upon concentration. It depends upon the difference in the concentration of this fructose ions from the luminal side towards inside of the cell, and this transport is facilitated by this transporter, this glucose transporter 5. Here it depends upon the concentration gradient of the sodium ions. It depends upon the concentration gradient of sodium ions and is also mediated by this transporter. So we have the involvement of sodium ion, then we have the involvement of this transporter, this sodium glucose transporter, and here we have only the facilitated diffusion. It does not depend upon the energy, it does not utilize the energy, it is simply the diffusion process. Here the movement is against the concentration gradient because here we have the concentration of the glucose molecule is more as compared to the luminal side, but this against concentration gradient movement occurs by the utilization of energy, and this energy is spent by this sodium potassium pump, and this energy utilization creates the sodium ion gradient. Here we have the less ionic concentration and here we have the more ionic concentration. This ionic concentration gradient of the sodium ion is used to transfer this glucose molecule and galactose molecule from the luminal side towards inside of the cell. This is how the absorption of these monomers occurs in the intestine. You know, this absorption of these carbohydrates occurs in the small intestine. So we have this involvement of these sodium glucose transporters and this glucose transporter 5 and glucose transporter 2. And remember, it occurs in 2 steps. In first step it crosses the apical membrane, and in the second step it crosses the basolateral membrane. To cross this apical membrane we have 2 transporters. One is the sodium glucose transporter. It transports the glucose and galactose from the luminal side towards inside of the cell, And to transport this fructose, we have separate transporter. We have glucose transporter type 5, and it transports the fructose from the luminal side towards inside of the cell. Transport of this fructose is a facilitated diffusion. It does not involve the expenditure of energy. Transport of glucose and galactose is secondary active transport because it depends upon the sodium ion concentration gradient. Here on the luminal side, we have sodium ion concentration is more as compared to inside of the cell. Decrease in the sodium ion concentration in the cell is maintained by the sodium potassium pump because it continuously transports this sodium ions from inside to outside of the cell. And this pump utilizes directly the ATP. This is the primary active transport, and here it is the secondary active transport. So this is all about the absorption of carbohydrates.